Mesa College, and I'm part of the Make Your Mark program. My project's on research on cerebral cavernous malformations. So to start off, cerebral cavernous malformations are a specific type of brain lesion that affect the central nervous system. They're mainly caused by the loss of function mutations in the genes CCM1, 2, and 3. The disease affects 1 in 200 of the general population, and out of those 1 in 200 people, 80% of them present the sporadic version, which is less intense, where they'd only have normally one lesion in their body, and 20% express the familial type, which is hereditary and is passed on by their parents, and they have they have a lot more lesions normally because of the mutation. And CCMs can cause a high risk of brain hemorrhaging, long-standing seizures, vascular thrombosis, and severe neurological deficits. And with all that, there aren't any pharmacologic therapies as well. The only way to treat the disease is by invasive surgery, which most of the time might not be possible because of the location of the brain lesion. And you can see on the left here is a graphic of what a CCM looks like. It's like a little raspberry, basically, of vascular growth. And you can see an MRI as well up here of the graphic, and down here of someone else with a lot more lesions in their brain. The only way that you can actually see whether or not a person has these is through MRI scanning, so it's a bit difficult to see since it's also misdiagnosed as cancer sometimes. Right now, the mechanisms behind CCM formation aren't currently known. As you can see here in a coronal section, there is a quiescent CCM here. Normally, the person that has these is asymptomatic, and through causes that we don't fully understand, they become exacerbated. And those mechanisms that influence the CCM lesions normally become exacerbated and can cause brain bleeding, thrombosis, and further lesion growth, like I said before. And it leads to disabilities in the patients mainly. What we're studying in the lab right now is to figure out what the propensity for CCMs to form in the central nervous system and why it happens. And the way we do that is through the PDCD10 knockout mouse model. And how that works is we inject an animal at P1 with tamoxifen, and that essentially silences the PDCD10 gene, which is the regulator that causes the mouse to have CCMs, basically. So we take the brains at P15, P50, and P80 for gene expression and histology. And those stages have been named the acute stage, progressive stage, and the chronic stage. And as you can see here, here's the difference between a control mouse of a PDC10 flux model and a PDC10 knockout model. The brain here, you can see there's lots of red, lots of lesions, and as it goes on progressively, the disease gets worse. And with the control brain, you can see that it stays relatively the same along their entire lifespan. And at least for the research that I did in the lab, I primarily focused on endothelial cells and astrocytes and their involvement in the disease. Astrocytes play a very large role in the disease because they secrete a protein called VEGF, which is called vascular endothelial growth factor. That protein basically starts the disease. No, it doesn't start, sorry. It basically just worsens the disease and causes it to happen with the lesions. They're critical for our study because both are involved in the gross endothelial changes that lead to the formation of CCMs, because when astrocytes release VEGF, then it causes the endothelial cells in the body, in the brain to become, lesionated. And as you can see here on the left, here is a staining of GFAP and IB4, which are two proteins that bind to astrocytes. The red is the astrocyte binding protein, and the green is the endothelial cell binding protein. You can see how the astrocytes are co-localized around the lesions, and how that only further suggests that they're a part of the whole lesion creation process. Astrocytes in particular play a critical role in CCM hypersensitivity to blood vessel formation by secreting VEGF. And this is relevant to explain the propensity of CCM lesions to form in the central nervous system, because astrocytes are primarily found in the brain. And from that, with the VEGF secretion, astrocytes integrate a circuit in neurovascular dysfunction during CCM disease, where they basically cause a feedback loop where they release the protein and then they cause damage to the CCM endothelium. And this is all in the knockout model as well. If there isn't the knockout, then it won't happen. And basically how we study this is by crossing our experimental model, the PDC-10 FlexLox model, with our ALDH EGFP line, because these have tagged ribosomes that when we harvest the brains at P80, we can see here that the isolated brain endothelial cells are green, as well as we have any EGF-mediated selection of astrocytic ribosomes, which we can both harvest and use for RNA sequencing. And with that, we can do, we can find out which genes are upregulated and downregulated in the disease to find further insight as to what the real causes are. And at least for the majority of my time at the lab, I spent it working on the in situ hybridization experiment to identify the role of astrocytes during CCM pathogenesis. And what the in situ hybridization is it's a experiment that qualitatively determines whether a select gene is being expressed via fluorescence. Currently, we're optimizing the experiment right now, but these are from a different experiment where we were testing for the presence of the EGF gene in CCM positive tissue. And through here, you can see these black dots. They're all markers for the EGF, so you can see that it's being expressed. And since there's lesions here, this is CCM positive tissue. And like I said before, this is GFAP, which is an astrocyte marker, and IP4, which is endothelial marker. And you can see how they're co-localized with each other. And the fact that the EGF is being expressed, right, localized on the astrocytes in an area where CCMs are being expressed, that can suggest more that the EGF and astrocytes play a critical role in this. And we primarily aim to take this information gained from the experiment and apply it to further research the mechanisms behind TCM and possibly find a pharmacological treatment for the disease. And mainly, through the in situ, there is a lot of preparation that needs to go into it, because the probe that you prepare through the RNA sequencing that you did before, you need to, or sorry, not the probe, just the samples that you've collected from the RNA sequencing, you need to prepare them as a probe, which is a very time-consuming task because you need to transform it into an RNA probe that there's just a lot of steps that take several weeks to do it. And we're at the step right now where we're just trying to optimize this experiment so we can further understand how the disease works. And I'd like to give acknowledgements to everybody in the lab that I worked with, my PI, Miguel Lopez Ramirez, and my other mentor, Monica Guma. And thank you.
Thank you so much, Iman. That's a fantastic talk. I'd like to open the floor to questions. So if you have a question, please use the raise hand function. Okay, we have a question from Alisa.
Hey, Elon, great job. That was a really great talk. I'm just curious, can lesions happen throughout the nervous system as well if they're like linked to astrocytes and that kind of thing?
Yeah, they can happen in the spinal cord, anywhere in the central nervous system where astrocytes are present.
Gotcha. That's really interesting. Thank you.
Mm-hmm.
Wow, that's super fascinating. Thank you. I did have a question on, so it seems like creating this probe is a whole ton of work.
Yeah, it'd take a while to explain it, so I just needed to simplify it a lot.
Absolutely. I just wanted to know what was, I guess, your most memorable experience in working in lab and learning how to create this probe.
I guess mainly just learning how to do all the lab techniques related to preparing everything, because every step of preparing it had another thing that I needed to learn. Like I did learn how to do pipetting, I needed to learn how to run multiple PCRs with different procedures, and then I had to transform the probe into into a plasmid so we could transform it into an E. coli, E. coli culture so we could get more of the cells with the DNA in it. And there's just so much that I've learned from it, and I think that's the main thing, because every step was something new.
Wow, that's an incredible amount of work. Thank you.
Mhm.
And I see another question in chat. Someone asked, or Bliss asked, Bliss, if you want to unmute yourself.
Hi, Elon. Great talk. I was wondering if you could explain the ALDH crossing with the PDCD10 just a little bit better.
So the way that it works is that we cross the two lines together, and because of that, it tags the ribosomes inside of the experimental line by crossing those two lines together, and it also allows us to we're able to select those astrocytic ribosomes and take them for RNA sequencing.
Awesome. Thank you.
Mm-hmm.
Great. Thank you again, Ilan.
All right. Thank you.